only do we get an ATP/CTP out of succinyl-CoA synthetase, but we're also fueling the respiratory chain, so we power ATP synthase. So we're going to get lots of ATP during the fed state, and I hope that makes sense. So all four, not only four, but in general, these molecules that I've written on the right here are high energy charge. Those things will be associated with the fed state. And so in the fed state, we are in a condition known as high energy charge. Okay, well, let's think about the fasting state. Well, in the fasting state, we're going to assume that we've been burning the ATPs, right? We're contracting our muscles. Recall that myosin, the protein in muscle that's responsible for muscle contraction, for every contraction it does, for every power stroke, it burns an ATP. Plus, on top of that, we're using neurons, like we're firing neurons, and to replenish the neuron concentration of sodium potassium on the right side of the axon, we're using sodium potassium ATPases. So there's lots of processes by which we're burning ATP, and at the same time we're burning our NADHs by NADH dehydrogenase, and so, we're wasting our acetyl-CoA inside the TCA cycle. So all of these low energy charged molecules tend to build up. Most of the ones I have written here, they're NAD, and then we also have our nucleoside diphosphates, nucleoside monophosphates, and so forth, and then we can sometimes also consider coenzyme A. So these molecules can be associated with the fasting state. Now, let's think about the fasting state, right? That's what we'll consider first, because those are the conditions under which we're going to run amino acid catabolism or amino acid degradation. So let's think about that. Let's think about our blood glucose levels. During the fasting state, we're assuming that we haven't had a good meal in a while, right? So our blood glucose levels are going to drop, right? So there's going to become less and less ability to run purely on glycolysis, right? So what we're going to do is we're going to switch over our metabolism a higher percentage of beta oxidation and amino acid catabolism, just because there's lower blood glucose, so there's less ability to run on the glucose, right, because we haven't had a meal. Now, this particular video and this particular picture is focusing on two amino acids. Those are glutamine and glutamate, okay? And what I want to, in separate videos, we're going to go over the mechanisms of glutamine synthetase, glutaminase, and glutamate dehydrogenase. And specifically in the catabolic direction, what I want you to understand is we're running from the direction of glutamine, shown on the right, and we're going towards the direction of alpha-ketoglutarate. And to understand why that is, let's think about the low energy charge, right? We have these low energy molecules, NAD, AGP, AMP, right? They're low energy, and so you can imagine that if we have low energy, low ATP, and low NADH, we're going to want to make more of those, right? Because we're assuming that NADH, ATP, and so forth, those are low, right? So we're going to want to make more of those. So we're going to run first from the direction of glutamine to glutamate. And the enzyme that accomplishes this is glutaminase, okay? And glutaminase is an allosteric enzyme, and it actually is stimulated by adenosine diphosphate allosterically. And I think that makes sense, right? That makes sense because ADP is associated with low energy charge. So if you need to make more energy by going towards alpha-ketoglutarate, it would make sense that a low energy charge molecule would turn on glutaminase allosterically, right? And it turns out that adenosine diphosphate is an allosteric stimulator of glutaminase. And glutaminase is a hydrolase enzyme, and it's going to convert glutamine into glutamate. Okay. Now, glutamate dehydrogenase, which is the next enzyme, is also an allosteric enzyme, and it's a reversible enzyme, but we're only going to focus on the direction in which it runs from glutamate to alpha-ketoglutarate. Now, one thing before we go into this reaction that I want you to see is when we're talking about alpha-ketoglutarate, recall that alpha-ketoglutarate can be shunted into the TCA cycle, right? And in the TCA cycle, it's going to ultimately get consumed by the alpha-ketoglutarate dehydrogenase complex, in which we form NADH. And then that's going to form succinyl-CoA, and we're going to continue that molecule on throughout the TCA cycle, and we're going to pick up, at least till the end of the cycle, one FADH2, one GTP/ATP, and one NADH. So ultimately, if we're going to the left on this graph, if we're going to the left towards alpha-ketoglutarate, what we're doing is we're making more energy. And I hope that makes sense. We need more energy because we're in low energy charge. We have lots of NAD present, we have lots of ADP, AMP, and so forth, but we have low NADH and low ATP. And remember that ATP is necessary for life. If you were to run out of ATP, you die. And that's really because muscles need ATP to contract, and neurons need ATP to fire. Okay, so if we're going to the left, that means we're generating more energy, and so the way we do it, at least through these two amino acids, is by converting them ultimately to alpha-ketoglutarate and shunting back into the TCA cycle. Now, glutamate dehydrogenase is technically a reversible enzyme. Technically it's reversible. But at least in mammals, only one of the directions is really observed. And that's the direction going from glutamate to alpha-ketoglutarate. And to understand this, especially during times of catabolism, we need to understand that, remember that glutaminase is stimulated by high levels of adenosine diphosphate. So we have to remember that glutaminase would be constantly turning glutamine into glutamate. And so by Le Chatelier's principle, glutamate dehydrogenase is going to be shifted towards the production of alpha-ketoglutarate and ammonia. Okay? So glutamate will go from the direction of glutamate to alpha-ketoglutarate. So the reverse reaction going from alpha-ketoglutarate to glutamate is not really observed. and especially during times of amino acid catabolism, it is certainly not observed. And glutamate dehydrogenase is also an allosteric enzyme. It's stimulated by high levels of ADP, and that makes sense because if we're in low energy charge states, we want to make more energy, and one of the ways we make more energy is by running this reaction to produce alpha-ketoglutarate, which goes into the TCA cycle to produce more energy. And it would also make sense that this enzyme is inhibited by high levels of GTP. So if we have high energy charge, GTP tends to inhibit this enzyme, and that makes sense. And that also brings into light why the reverse reaction is not really observed. So let's say we're in high energy charge, so we have high levels of NADH and high levels of ATP and GTP and so forth, right? So we have high levels of those. Well, why wouldn't the reverse reaction going from alpha-ketoglutarate to glutamate occur? Well, if we have high levels of GTP, this enzyme should be allosterically turned off, right? We're either going to be in low energy charge or we're going to be in high energy charge. So if this enzyme is turned off, even if we have lots of alpha-ketoglutarate present, it's not going to reverse itself. So when GTP is present at high enough, or we can say high energy charge is present, this reaction will not simply just reverse itself. It's going to be allosterically turned off. But when we're in low energy charge, when we have high ADP present, this enzyme will go from the direction of glutamate to alpha-ketoglutarate, partly because we're building up glutamate from glutaminase, and by Le Chatelier's principle, it's going to force the reaction to produce alpha-ketoglutarate. Now, one thing I want to point your attention to in this reaction sequence of going from glutamine to alpha-ketoglutarate is in both of these reactions we're producing ammonia, okay? And remember how what we talked about in the urea cycle video, that high levels of ammonia are not good. They're toxic, especially for brain cells. So remember that ammonia will be activated by carbomoyl phosphate synthetase 1, and then that carbomoyl phosphate will be shunted into the urea cycle, and the ammonia will ultimately be excreted in the form of urea. So when we're metabolizing a lot of amino acids, especially these, we are producing ammonia, and it will eventually be excreted as urea. So I hope this video gave you a little bit of intuition on energy charge in the cell. Remember that when we're in high energy charge, that's going to basically favor biosynthesis, and we're not going to talk about biosynthesis here. We'll come back to that in the biosynthesis playlist, and we'll actually use this exact same picture. But we're talking about catabolism, and in the direction of catabolism, that's going to be favored when we're in low energy charge. We have low energy molecules, we need to make higher energy molecules. So we're going to favor the direction that goes from glutamine to glutamate, and then from glutamate to alpha-ketoglutarate. And make sure you understand the allosteric regulation of these enzymes. See you in the next video. Amino acid catabolism. And we haven't really gotten much into the catabolism yet. We've talked a little bit about transaminases. We've mentioned the urea cycle. We haven't really got into the catabolism yet. And we're still not going to get there for a few videos. But what we're going to do in this video is we're going to talk about the glucose-alanine cycle. This is a very, very important cycle, especially for muscle cells that are contracting anaerobically. Okay, so a lot of times when muscles are actively contracting, they're doing so without oxygen. In other words, they're not using their electron transport chain. So they're contracting anaerobically, so they're having to run on glycolysis. Okay, so we're going to mention in this video why that becomes important and how we counteract it. Okay, so one thing we have to understand is that there's a constant sort of tug of war between muscle protein synthesis and muscle protein degradation. So it's a very dynamic thing. It's not like you have muscle protein and it just stays there statically. No, you're constantly degrading muscle tissue and you're constantly synthesizing muscle tissue. And let's say that your muscle synthesis exceeds your muscle degradation, that would be hypertrophy, right? And if your muscle degradation exceeds muscle synthesis, that would be atrophy. So it's a constant tug of war, and ordinarily if you're eating enough protein and you're not working out, that would be more of a perceived static state, where you're not actually degrading more than you are synthesizing. But at any given time, you are going to be degrading muscle proteins, and those muscle proteins are going to get degraded to amino acids. So our proteins are going to get degraded to amino acids. And just like any amino acid, right, they have an alpha amine group, right? And that alpha amine group can get transported into the alpha amine of glutamate, right? It can get transported into the alpha amine of glutamate, okay? And that's important because glutamate, as we've seen, is important in the reaction of transaminases. So what we're going to do right now is we're going to talk about the part of the glucose-alanine cycle that takes place in the skeletal muscle. Okay. So what we're going to have at first is we're going to have pyruvate, right? We're going to start with pyruvate, right? And what's going to happen is we're going to have the reaction of alanine aminotransferase. So I'll put AA. Actually, let's not do that because I already used that for amino acid. Let's, my mouse works. Let's do, instead, let's say ALA-A, or alanine alanine aminotransferase. Okay, so this is the reaction of alanine aminotransferase. And pyruvate is essentially going to accept the amine from glutamate, right? And so it's going to generate alanine, right? And so the glutamate is going to come in, transfer the amine group effectively, and what you're going to get out is alpha-ketoglutarate, right? So you're going to get alpha-ketoglutarate, and that alpha-ketoglutarate, as we've seen, can then go into the TCA cycle, right? Okay, and of course that TCA cycle is in the mitochondrial matrix, right? Well, we mentioned that alanine is a very important transporter of amines in the blood, and to understand that, let's actually look at the structure of alanine. So alanine is going to look like this, and notice that on the alpha carbon, it has an amine group. So that alpha amine is going to be this right there. And in fact, that amine came from the alpha position on glutamate. Okay, so that was on the alpha position of glutamate. But one of the functions of alanine in the blood is to transport amine groups. And one of the reasons this is important is because, as I hope you've found in some of the other videos, is that ammonia is toxic, especially in large amounts. You don't just want free ammonia floating in the blood. So one of the ways that we get around doing that is we attach it to alanine, and this is catalyzed by alanine aminotransferase. We take the alpha amine from glutamate and transport it on pyruvate in the form of alanine. So having said that, before we go any further, one of the ways that we transport amines in the blood is alanine. That's the one we're going to discuss first. One we'll do second is going to be another transporter of ammonia in the blood, and that's going to be glutamine. There's a huge difference in the transportation of the amine. The transportation of the amine in alanine is on the alpha position. So this position right here, this is the alpha position. However, in glutamine, the amine is transported in the amide linkage that's on the R group of glutamine. The alpha amine of glutamine is not going to be the direct amine donor of glutaminase, which we already have mentioned to an extent. So the actual location of the amine is different, and I hope to elucidate that for you soon. Okay, so now we're talking about alanine. And alanine can actually move through the blood. So alanine will move through the blood, and it's going to go to the liver. Right? Alanine is going to go to the liver. Now, effectively what the liver is going to do is it's going to reverse this process. It's going to reverse this process of the transamination. So right now we have alanine, right? And it's going to reverse the process. So we're going to require an alpha-ketoglutarate, right? Alpha-ketoglutarate. And we're going to get out a glutamate, right? And in the process, we're going to generate pyruvate. So we're going to generate pyruvate. And again, this is catalyzed by an alanine aminotransferase. Okay, so what have we seen? Alanine goes in the blood to the liver, and alanine aminotransferase there is going to convert it back to pyruvate. What I want to mention at this point is that the actual transamination that's shown here from alanine to pyruvate, this particular reaction occurs in the cytosol of hepatocytes. Remember that liver cells are also referred to as hepatocytes. Now, in the hepatocytes, the glutamate that's produced in the cytosol through this reaction can enter the mitochondria, and we will discuss the fate of glutamate in a few minutes. But suffice it to say, it will react with glutamate dehydrogenase, forming either an NADH or an NADPH, and then it will generate alpha-ketoglutarate, which will go into the TCA cycle. Okay, now we have pyruvate in the liver. Now, in order to get glucose back to the skeletal muscle, gluconeogenesis is going to occur. If you need help with gluconeogenesis, certainly go review the playlist on that. But suffice it to say that pyruvate is going to be converted back to glucose. Pyruvate is going to be converted back to glucose, and that process is gluconeogenesis. Right? Remember, in the mitochondria of the hepatocytes, remember that the pyruvate is produced by the transaminase, which first goes to the mitochondria, and there it's going to react with pyruvate carboxylase, right? And that's going to form oxaloacetate. And from there, then we can do various things. We can either convert it from phosphopyruvate by phosphopyruvate carboxykinase, and then transport PEP back out into the cytosol where gluconeogenesis can occur. Or we can take oxaloacetate, react it with malate dehydrogenase in the mitochondria to get malate, transport malate across the membrane, right? And then we can use cytosolic malate dehydrogenase to convert it back to oxaloacetate, and then we can use cytosolic phosphopyruvate carboxykinase to give us PEP, and then we can use gluconeogenesis on that. And if you recall from the previous videos on the aspartate margin aminosuccinate shunt, we found a third way to do this. We can essentially convert oxaloacetate into aspartate, and that's done by aspartate aminotransferase, and then we can transport aspartate through the membrane of the mitochondria, and then use cytosolic aspartate aminotransferase to get it back to oxaloacetate, and then we can do gluconeogenesis. But the whole point in saying this is that this process right here is going to be gluconeogenesis, and we're going to end up getting glucose back. Now, the glucose that is produced is going to essentially leave the cytosol of the hepatocyte, and it's going to re-enter the bloodstream. So that glucose is going to re-enter the bloodstream, and it's going to go back to the skeletal muscle, right? So now we're back on the skeletal muscle side, and the glucose that's made is going to be converted back into pyruvate. And what's the process that does that? Well, that's the process of glycolysis, right? That's the process of glycolysis. And one of the things that you should realize about muscles that are actively contracting anaerobically is they're largely running on glycolysis. And if you have a tissue like a skeletal muscle that's actively running on glycolysis, you must constantly feed it glucose, right? But if you're not getting glucose from the diet, in other words, you're doing amino acid catabolism, you're not getting glucose from the diet, so you have to get it from somewhere. So one of the ways that you supply the skeletal muscle with glucose is by doing this glucose-alanine cycle, okay? That's very important to understand. If you're not getting glucose from the diet, you have to get it from somewhere. It's done through this cycle. So I hope this cycle made a little bit of sense. Okay, so now before we go into glutamine metabolism, what I want to do is I want to talk about this glutamate that we generated right here. What happens to that glutamate? Well, the glutamate, like we said, can be transported into the mitochondria, right? So we have our glutamate that's in the mitochondria, right? And the glutamate can react with glutamate dehydrogenase. And remember that glutamate dehydrogenase is technically a reversible enzyme, but under normal conditions it only runs in this direction, so that's why I'm drawing it on my arrow. And in this reaction, what we're going to do is we're going to use nicotinamide adenine dinucleotide, or the phosphate version of it, right? And then we're going to get out either NADH or NADPH. This enzyme is unique in the sense that it can actually react with either NAD or NADP, and that's important to understand. And then in the last step, we're going to do a shift-based hydrolysis and cleave off ammonia. So that ammonia will then go into the urea cycle. And in the process, we generate this very special product known as alpha-ketoglutarate. And like we mentioned before, that alpha-ketoglutarate can then go into the TCA cycle, and that's the enzyme that consumes it directly. Well, it's the alpha-ketoglutarate dehydrogenase complex. Okay. So now we've seen what happens to the glutamate, but we have one other amino acid that we need to deal with in terms of transporting amines in the blood, and that amino acid is glutamine. Okay, so one of the things that can happen is when you have glutamate present and amines, there's a reaction that can take place, and we're not going to do the mechanism here, but we'll do that in another video. What can happen is if we need to transport amines in the blood, and we have excess amines, what we can do is instead of putting the amines into the urea cycle, we can actually ligate them to glutamate. So this is important. Whenever you have excess amines, or in other words, excess ammonia, remember ammonia is toxic to animals. So instead of just having the ability to funnel it into the urea cycle, we can actually ligate it to glutamate. Okay? And actually, why don't I do this? Instead of just drawing glue, let me actually draw out the structure just so you can see what's happening. Okay? So here's going to be our glutamate molecule. Okay? This is our glutamate molecule. And this irreversible reaction, number one, is going to burn an ATP. It's going to burn an ATP, right? And then what it's going to do is it's going to put the ammonia in, right? And it's going to ligate it to glutamate. And in the process, what we're going to do is we're going to generate glutamine. Right? And so one thing I mentioned earlier is it's not actually the amine on the alpha position that we're transporting. It's actually the amine that I'm going to do in red here. It's this amine. It's the amine that's tied up in the amide linkage on the R group of glutamine. So this molecule right here is L-glutamine. So we're actually transporting the amine on the amide linkage of the R group. And so that's one of the ways in which we can transport amines in the blood. Or we could say transporting ammonia in the blood is tied up on the R group of glutamine. Okay. And one thing that we can do also when we get glutamine to the liver is we can further react glutamine. We can react it with glutaminase. So the enzyme we're going to react this with is glutaminase. This is a hydrolytic enzyme that's going to hydrolyze off the amine, and you're going to regenerate glutamate, right? And of course, in the process, you end up generating ammonia, right? Glutaminase is a hydrolase enzyme. Let me put the water here. Glutaminase is a hydrolase enzyme that's going to cleave off ammonia, and in the process, we regenerate glutamate. And this enzyme, glutaminase, is expressed at very high concentrations in the mitochondria of the hepatocytes, the liver cells. So when you get the glutamine inside the liver, it will be transported through the mitochondrial membrane into the mitochondrial matrix, where glutaminase will react with it to form glutamate. And then that glutamate that's made is going to follow the same physiology that we saw before. It's going to react with glutamate dehydrogenase, and again, we've seen this reaction before, I showed you it before, and it's going to give you alpha-ketoglutarate, and it's going to give you either NADH, or it's going to give you NADPH, because the enzyme can react with NAD+, or NADP+. And I might also mention that the enzyme that ligates the ammonia to glutamate, this enzyme has a special name. It's called glutamine synthase. Glutamine synthase. One key is that it is ATP dependent. Okay, it's an ATP dependent reaction, and so what we end up doing is we end up putting the ammonia on glutamate and make glutamine, and that's how we transport the ammonia in the blood. Okay, because remember, ammonia is toxic at high concentrations in the blood. It's toxic. We don't want ammonia just running free. In fact, it's very damaging, especially to brain cells. So the two main ways we get around transporting it are we attach it to pyruvate to make alanine, or we attach it to glutamate to make glutamine. And just keep in mind that we're transporting it at different positions on the amino acid. In alanine, we're transporting it on the alpha carbon. On glutamine, we're transporting it on the amide linkage on the R group of glutamine. So I hope this video gave you a little bit of information on how we transport ammonia in the blood. See you in the next video. Welcome back to the playlist on amino acid metabolism. In the last video, what we did is we looked at the glucose-alanine cycle, and we also looked at generally how ammonia is transported in the blood. And we saw that it's done by ligating the ammonia ultimately to pyruvate to make alanine, or to glutamate to make glutamine. And the enzyme that made glutamine was called glutamine synthase. And what we're going to do in this video is we're going to look at the organic mechanism of glutamine synthase. And actually throughout the next few videos, we're going to look at the mechanism of several of these enzymes, including glutaminase, glutamate dehydrogenase, and some of the transaminases. Okay, so let's look at the mechanism of glutamine synthetase. Now, this mechanism involves ATP. That's why it's referred to as a synthetase. It's ultimately going to be a condensation between glutamate and ammonia. So it's a condensation reaction that requires a nucleoside triphosphate. That's why it's referred to as a synthetase. So also keep in mind that this is a magnesium-dependent enzyme, and the magnesium is used to stabilize the incoming ATP molecule. Remember that magnesium has a 2+ charge, phosphates are negatively charged, so that stabilizing effect sort of holds the ATP in close proximity, so the glutamate lone pair can do a nucleophilic attack on the gamma phosphate of ATP. So this right here, this is the gamma phosphate, and the first step of the mechanism is going to be nucleophilic attack on the phosphorus atom, and that's going to generate a trigonal bipyramidal intermediate that can be seen in the next picture. So we have this trigonal bipyramidal intermediate that's still stabilized by that magnesium ion, and in this step of the mechanism, what's going to happen is, I can draw my lone pair,